Yeah, welcome everyone, people in person and online. So we're very grateful to have Linnéa Gyllingberg today to give us a talk. I'll just read her bio for you. So Linnéa Gyllingberg recently received a PhD in applied mathematics from Uppsala University within the Center for Interdisciplinary Mathematics in Sweden. In September, she will start a postdoctoral scholar as a postdoctoral scholar at the Department of Mathematics at the University of California in Los Angeles, USA. It sounds very exciting, with Professor Mason Porter. Her research focuses on developing and analyzing mathematical models and models for different biological applications. She uses dynamical systems, network models, and agent-based modeling, as well as statistical methods to understand collective behaviors, decision-making, and complex systems. So all sounds very, very interesting. So I'll hand over to you, Linnéa. Thank you.
Thank you very much for the introduction, Amy. So it's very nice to be here, or not to be, I mean, I'm at home, but to present to you in South Africa. And so the title of my talk is Mathematical Models of Basal Cognition, which is one project I've been working on lately. But I'm also going to talk briefly about some, wait, the sound is really weird for me, like it's echoing a lot. I don't know if I should.
Oh, maybe, let me just check if there's anyone.
Is it like mutable microphones?
No, everybody is. Is this better if I mute this?
Yeah, or now I don't hear you. You talk? Are you?
Yeah.
Okay, I'll survive, I think. But yeah, so my main focus now is on mathematical models of basal cognition, but I thought I'd first just talk briefly about some other projects I've been doing throughout my PhD. And one thing that I've been looking at is ecological models. So I have this paper called Finding Analytical Approximations for Discrete Stochastic Individual-Based Models of Ecology. That's a really long title, but what we basically do in that paper is that we look at a class of individual-based models, so model that is defined by individuals interacting, in this case on a lattice. And this model reproduces very interesting spatial dynamics. So when we simulate it, we get interesting spatial dynamics depending on the parameters. And we ask if we can find analytical approximations of these types of models. And the answer to this is yes. So we derive like 3 different analytical approximations depending on the parameter regimes to study this type of model. And in another paper, I've also looked at models of collective behavior. So this is a paper called Using Neuronal Models to Capture Burst and Glide Motion and Leadership in Fish. So normally when you look at fish schools, they move like as this group, right? They have this pattern of movement, which is very well described by something called self-propelled particle models. So it's models where you assume that the fish look around them and move with constant speed and just update their direction depending on the fish around them. And those were great for big schools, but if you look at how fish actually move, they move with something called burst and glide movement, meaning that they burst like this. Can you see my mouse? My pointer?
No. Yes, we can.
Okay,
we can,
perfect.
yeah.
Yeah, perfect. Yeah. So they move with this. So they burst for a bit, accelerating their motion, and then they glide, and they burst, and they glide. And this is not incorporated in most models previously. So what we did was that we thought of taking inspiration from a model of neuronal interaction. So this is the Fitzhugh-Nagumo model, which models how the action potential in a neuron is transferred between neurons. And it's this 2-dimensional dynamical system. And then we alter these equations and think of it as like, what if we model the speed of the fish and we link their speed according to some function? And what we get is that we get quite complex swimming dynamics. So we can actually capture a lot of swimming dynamics with this simple model, coupling it to some interaction function. We get different types of swimming dynamics and different types of leadership in these fish, depending on our parameters. So that was one other project. And then I'm also interested in more philosophical aspects of mathematical models. So in this paper called The Lost Art of Mathematical Modeling, we think more broadly about like, what do we actually do as mathematical modelers, and with a special emphasis on mathematical biology, because that is my field of study. And we would say that the challenges in mathematical biology can be roughly broken down into 3 activities. And that is formulating models, analyzing models, and fit or comparing models to data. I don't know if you agree with me, but I think that more or less that's what we do as people working in mathematical modeling. And even though it's not like a straightforward line that you start with formulating, then analyzing, then fitting, it's like a circle, you go around, you go back and forth, and adjust your model based on maybe the data and so on. But somehow this is the 3 activities. And when we formulate models, that could, for example, be like the Vic-Check model, which is the self-propelled particle model, or the Turing's work on morphogenesis, so how patterns form in like animals, or like SAR models, which I guess most people are familiar with now after the pandemic, or like adaptive dynamics. That's another type of models used in evolutionary game theory. So these are just some examples. And then when we analyze models, we might do like some sort of stability analysis. We look at bifurcations, or we look at the limit of large number of agents. So what's happened when we have many fish or many whatever atoms interacting with each other. And then we fit and compare the models to data. And that is like the emphasis in machine learning a lot. That is on the fitting and comparing models to data. And what we do in this paper is that we say, in rise of the rapid developments in machine learning, we should actually not just focus on activity 3, we should rather also focus in mathematical biology on formulating models. And we argue for this with something called integrative pluralism, which is the idea that like any biological system can be modeled in an infinite number of ways. So if we look at example at like fish movement, which was the focus on my previous paper, you can, for example, model fish. If you have a school of fish, it works quite well with the self-propelled particle models. But if you have like one fish swimming, they swim with this burst and glide movement. So there are models for this as well. And that burst and glide movement can be modeled through a hydrodynamical perspective. So thinking of that the reason the fish do this is some sort of conservation of energy. And then you have this PD models of this type of swimming dynamics. Or you can have a behavioral ecology perspective. So when fish move with burst and glide, they see more prey, and you can model this in a different way. Or you can think about this as social dynamics and decisions in pairs, which was what I did in my previous paper, that is looking at like, how can we model this, thinking of that the fact that they interact with each other when they swim like this. Or you can model the neurobiological aspects of it, like the activity in the brain, how that affects the motion. So just with one very specific thing, modeling fish motion, we have tons and tons of different models. And what I would say is that this pluralistic approach says, like, depending on what questions we want to answer, the model used is different. And these models can contradict each other, but that's not a problem. We shouldn't try to reduce this to just one model that can explain everything, because having more models helps us understand more. And then we also ask, like, why do we even need mathematical models? Can we just like replace everything by, for example, machine learning or something like that? And there we argue, I really like this picture this is from a paper by Brial et al. Called Explanation and Organizing Principles in Systems Biology. Where they talk about phenomenological versus mechanistic models. So if we look at these, if we look at this figure, we see these data points, and then we see a line, and all these different models, like this y of t equals a times cosine of t, and this model b, which is a differential equation, or this model c, they all represent the data. But model A is just curve fitting, and that is great for prediction, whereas model C also includes a mechanistic description. So having these types of mathematical models actually gives some sort of understanding of the underlying system. And biological systems are more complex systems, meaning that they're more than just the data. And data itself is not understanding. Understanding requires a conceptual framework, a theory that identifies the fundamental variables and their causative influence on each other. And even though models can be very useful for prediction, models can also be used to guide data collection for future experiments, capture qualitative behaviors of overarching interests, or like suggest dynamical analogies, or lead to new scientific questions being posed. So there are so many different reasons to do mathematical modeling other than prediction, and that is what we argue for in this paper. So these are quite diverse topics, but I somehow, together with like what I will talk about, the main focus of my talk will be, I managed to like combine it into one thesis by thinking of like the biological systems that I study, focus a lot on like oscillations and feedback, and then I take like a bigger picture on how to do mathematical modeling and mathematical biology, and thinking of what is the art of it. And I combine it into one thesis called The Art of Modeling Oscillations and Feedback Across Biological Scales. So this was just a background of what I've been doing before. And now I will talk about the main topic, which is basal models, mathematical models of basal cognition. And what I really want to talk about is this organism called the Physarum polycephalum, or the slime mold. So it looks like this. It's this yellow. Has anyone seen this, heard about this organism before, by the way? You can raise your hand and I can see you. Oh, a few of you. At least Francesco and Amy, and someone else who I don't know who it is, but nice. So the slime mold, oh, now I should have removed this. You don't need, no one knows Swedish, so this is irrelevant. But it's called if you translate this Physarum polycephalum, it means like the multi-headed slime mold. In Swedish, it's called swamp, meaning it's like slime mushroom. So that's why I'm saying it's not a fungus, because it's in Swedish, you would assume it's a fungus. What it rather is, it's a cellular or a unicellular organism, meaning that it's just one giant cell with multiple nuclei, so tens of thousands of cell nuclei. And this organism forms a network of tubes where nutrient is transported. So here we see like a picture of it zoomed in, so you can see these networks in the slime mold, and here you can actually see this tube, and here it's even more zoomed in, and you can see the individual cell nuclei. So these are cell nuclei that are stained by some protein, I think, so they you can see them. But why I'm so interested in slime mold is that slime molds can actually learn. And here I define learning as a change in behavior evoked by experience. And one way they show learning is the simplest form of learning, which is habituation. So habituation means that like every morning when you put on your clothes, you will in the beginning feel your shirt on your arm. But after a while, I mean, having that information constantly, the feeling of your clothes, that's unnecessary information. So you get your senses get, or your they get habituated to this feeling, so you won't feel it all the time. And that is basically what habituation is. And this is an experiment where they demonstrated habituation in slime molds. So they have like a slime mold here and some food. I think this is egg yolk, because they like egg yolk. And then they have this bridge where they put salt, and slime molds don't like salt. And then they put it here and it tries to avoid it. They take away the slime mold, and the next day you can see how it's habituated to the salt, so it just moves more quickly through the salt barrier. So after a while, it gets habituated. And slime molds, there are also experiments showing that the slime molds can anticipate periodic events. So what they've done in these experiments is that they have a slime mold here, some food here, and in between they have a lamp blinking. And slime molds, they don't like salt and they don't like light. So if you blink the lamp every 5 minutes, after a while, the slime mold learns the frequency of the lamp and will like retract when the lamp will be turned on. So it has this way of anticipating periodic events. And it can also make decisions and do problem solvings. So here is a maze, which is a representation of the Tower of Hanoi, actually, where they put a slime mold here with food and food here. And after a while, it solves this maze, meaning it finds this optimal path between the food sources. And even more impressively, the slime mold can share information. So one slime mold, which is just one giant cell, can combine with another slime mold, another giant cell. And if this one slime mold knows something, like it has learned the anticipation, the periodic events, it can transfer that knowledge to the other slime mold. And then they will like divide again, and they both will know the same things. Which is quite amazing, right? It's just like unicellular, just one giant cell, and it still can learn things and share that information. And one fun thing about the slime mold is that it can solve shortest path problems. So these Japanese researchers, they did experiments where they put oat flakes, because slime molds like oat, at the same places as the Tokyo railway system. And the slime mold model, after a while, form a network which was actually as cost efficient and robust as the real rail network. So one unicellular organism is as good as planning networks as like a bunch of engineers, which is quite amazing. So just to show how it looks like when it does this, this is a video of this experiment. So the slime mold kind of like explores the environment, and after a while it forms these shortest path solutions, the optimal solutions to transport the food. And then you can ask, like, why am I, as a mathematician, interested in this? Why am I interested in slime molds? Well, I'm interested in how can we model this behavior that it can solve shortest path problems. And this is being modeled usually by a network. So we think of the slime mold as a network. So for example, in this case, you will form a network like this, meaning that each crossroad will be a node, and then there's edges between. And what we model is then the thickness of the tubes between two nodes, I and J. And they changed according to some equation on the flow in this network. And there are multiple papers exploring this particular model, both from more mathematical perspectives, where they proved, for example, that these equation will always find the shortest path, and for more like engineering applications, can we use these algorithms to build some sort of to build algorithms, and or from more biological perspective. So this is not new. This has been done. This was like introduced in 2006, so soon 20 years ago. But what is new in the biological aspect of slime mold is that there's more and more evidence that oscillations. So if we look at this video, we see the slime mold, and you can kind of see, can you see how it oscillates? Like the tube changes like this all the time. So the tubes oscillate, and it also oscillates when it moves forward. So there's more and more evidence that oscillation plays a pivotal role in the learning in slime mold. And in this recent paper by Bossard et al., they say that we hypothesize that, as in the brain, modulation of spontaneous oscillations may sustain learning in slime mold. So the previous models haven't incorporated oscillations. So what we wanted to do is to think of like, can we incorporate oscillations into a model, finding these shortest paths, and see what happens if we have oscillations, and how can this maybe sustain learning? So this was like the background to what we wanted to do, understanding the role of oscillations in slime mold learning. And how do we do this? Well, we do this with something called a current reinforcement model, and I will explain to you soon why it's called a current reinforcement model. And the idea is that we model, again, the slime mold on a network. So in the network, we have nodes and we have edges. And we have three types of nodes. We have sources, sinks, and normal nodes. And depending, and at each node, we have a number of particles. So the particles can be the food sources. So we think about like the slime mold transports food. So it could be all these food particles or the cell nuclei or things that are transported in the tube of the slime mold. So we have a number of particles in each node. And then we have a length between the node, Lij, and we have the thickness of the tubes between two nodes, Dij. So this is how thick it is. So this node or this edge is thinner than this, for example. And then we have some input and output A and B, which we first define as constants. And then we have some equations governing how the number of particles at each node changes, and some equation governing how the thickness of the tube changes. And these are a bunch of equations, but what they actually just mean is that the more particle flow we have, the tube gets thicker, and the less particle flow we have, the tube gets thinner. And then one can ask, like, why is this called current reinforcement? Well, it is because there's some parallels between the Physarum, the slime mold network, and electrical networks. And this establishment was made already by Taro et al., the one who wrote this paper, which I first showed this model on slime mold network formation. And the idea is that, like, in an electrical network, the length in space would be the length in space in Physarum, whereas the amount of nutrient, the number of particles, is the potential of voltage in the network. And the flow of nutrient is the current in an electrical network. And the thickness of tube is conductivity, and so on. And what is reinforced in this previous model is this current or the flow of nutrient, because this is what is reinforced here. So that is why it's called a current reinforcement model. So it's based on this analogies to electrical networks. So what happens if we simulate this model? We can see that depending on the length, so here we just have an inflow of particles and an outflow of particles, and we want to see if the model will choose the flow with the shortest path. And what we see here is that it does. So we have like the thickness of the edges or the conductivity, it will go to 0 for the longer path, and then it will approach some value for the shorter path. And we also show that mathematically that this model have a stable equilibrium when this is the shortest path, and a stable equilibrium here when this is the shortest path. So this is just some mathematical results we have in this project. But what we were actually interested in was oscillations, like how do oscillations affect the formation of networks in this in the slime mold or in this type of models. So what we do then is we take the same model, but now we say that we don't have constant input and output. Instead, we have oscillating input and output, meaning that, so we defined it according to some function, so there could both be an inflow here and an outflow here, and an inflow here and an outflow here. And what happens when we simulate this? So in this case, this is the shortest path. So this is the shortest path, whereas this is the longer path between 1 and 3. And we get this oscillating number of particles at each node, and the conductivity, the thickness of the tube, is also oscillating. And the input and the output is also oscillating. And after a while, it self-organizes. So it's always a constant input and output flow in the model. And one way to understand this model more is to look at a bifurcation diagram. So to see how does this conductivity or the thickness of the tube change depending on one parameter. And in our case, this parameter is the phase difference. So the phase difference here is like the difference between, here is the phase of one node, and here is the phase of the other node. And we'll look at how these change. And what we can see here is that depending on, for example, when the phase difference is pi, we have one type of network that is formed. And when the phase difference is pi/2, we have another type, disconnected network. And basically changing the amplitude ratio and the phase difference give rise to multiple different networks. So what we've done here is actually studying a very basic network. I mean, it's just a cyclic graph with 5 nodes, which maybe doesn't make that much sense for a slime mold, because if we think about it, in the beginning, we had like all these very complicated network structures, like here, for example, or here. But this is because we want to understand this simple model as much as possible before we go on to studying larger networks. And that we have done, and I'm just going to show a simulation of what happens when we simulated this on a larger network. So this is these are the oscillating nodes, the red and the blue ones, and these have the same phase, and this one has another phase. And then now what we will see is like the color of the nodes indicate the number of particles. And then you can see how the edges of the network oscillates and change size, and also the number of particles oscillate over the network, but it transports between like this pumping nodes. And that is just like the first part of this project, and we have it as a preprint called Minimal Model of Cognition based on oscillatory and reinforcement processes. And this is joint work with QSUN, who's at Nordita, the Nordic Institute for Theoretical Physics in Stockholm, and David Sumter, who was my advisor for my PhD studies. But what I'm in general interested in, because I want to study basal cognition in general, and there's more and more evidence that a lot of different organisms without a central nervous system or even neurons at all show different types of cognitive abilities. So this image is taken from this paper, Reframing Cognition, Getting Down to Biological Basics in Philosophical Transactions B, which I think is a great paper. It's a special issue about different types of organisms that show basal cognition. And here we have the slime mold. There are some bacteria, some flatworms. And what I'm actually interested in is understanding the role of oscillations of this. So can we build some sort of more general mathematical framework to understand how oscillations affect minimal cognition? And for this research program, I recently received 4 years of funding from this Swedish foundation, Wallenberg Foundation. It's like the richest family in Sweden who owns all the banks in Sweden. But they also give a lot of money to research, which is nice. So I'm gonna soon move to Los Angeles to work with Mason Porter. He's a professor in applied mathematics, doing network science and dynamical system. And for more biological insights, I'm going to work with Audrey de Sautour. She has all the videos I showed you, or the video of the oscillating slime mold, that is from Audrey's group. So she's a slime mold expert in Toulouse. And I recently visited her to see her lab and see some real slime molds. And then I'm going to continue working with QSUN at Nordita. And yes, I know I was given 1 hour, but this is not an hour at all. I hope you forgive me. So thank you. That was all I had to say.
Thank you so much. Yeah, thank you, Linnéa. That was very interesting. Do we have any questions in person, and then I'll see online if there's anything.
Yeah, thanks for the very interesting talk. I was just wondering, in your simple model with the graph of 5 nodes, what was could you qualitatively think what the advantage was when you introduced the oscillations versus the no oscillations?
Yeah, that's a very good question. And so in these previous models, which has done before, they find the shortest path, but all the solutions are always like optimal spanning trees. So it's like this perfect solution. But the thing is, like in nature in general, and in slime molds especially, these type of spanning trees are quite rare in the network formation, because like the slime mold rather form loops. because it doesn't want, for example, if we take this example, we still want to have links with like less good food sources, because nature is always changing and you might not know what will happen in the future. So what happens when we have oscillations is that we get this kind of, in this very simple example here, we can see that like instead of just having a solution directly here, you still connect some points, even though it's like very thin connection, it's still connected, meaning that it forms like, for example, loops and solutions like that, which are seen in slime balls, but not seen in this previous model. So that is one advantage of having oscillation. And then you might think of like another advantage is like, which we haven't explored here, is like the information transfer. So how can information really be transferred in slime? And one hypothesis there is that it's like, it's actually, I mean, oscillations contain information in form of amplitude, frequency, and so on. So that is a form of like having information in the system instead of just having a static signal. You can have some sort of information. So when you anticipate periodic events and so on. Was that an answer to your question?
Yeah, thanks. That's very cool, because it makes me think that, like you say, in nature, maybe a graph that is dynamically changing its structure the whole time, the oscillations might be better to, cater to something that changes. It's not a static graph the whole time. So it seems like a nice method to update to its environment. Like, that's very cool.
Thank you.
Can I ask a question? Sorry, you showed us nicely that these unicellular things react to the environment, and they learn. Would you say that they are conscious?
No, I don't think so. But I don't know. That's a different question, and that's a good question, because I think it's important when doing this, and I've been reading a lot about this. There's this whole community doing minimal and basal cognition, and what they emphasize is like the difference between cognition and consciousness. Like having cognition doesn't mean that you're conscious necessarily. Cognition is a form of being able to learn, change your behavior based on experience. But I'm not sure I would say that they are conscious. I think that's another level of cognition. But yeah, I'm not the biologist here, so don't know for sure.
No, I was just asking provocatively, because where do you put we also learn from experience. But we are supposed to be conscious. Probably, where do you put the line? The line,
yeah, I mean, that's a super tricky question. I don't know where to put the line.
I don't.
But I think it's a very important question, and that's why I think it's so important to study these types of systems and organisms, because there's been, in neuroscience in general, there's been like this view of, well, if you have a central nervous system, that's when you have cognition. But like, that's not how evolution works. Somehow there's like evolution is continuous in the sense, okay, it can have jumps and so on, but it's still like, if you think about where would cognition come from, where would our brains come from, it needs to come from some other organism without a central nervous system or even neurons. So it kind of puts emphasis on that as well, that there is some sort of continuum of cognition and those, which I think is very interesting.
No, I know. I also don't know the answer.
Oh yeah.
Sorry.
Just like also in related to Francesco's question, in your future work, what's some would you have examples already of I'm wondering what the minimal tasks of cognition is that you're going to consider. What tasks for minimal cognition to study?
Yeah, I think I mean, that's a good question, and I would also because I'm interested in models, of course. So I'm interested, and going back to when I talked about like why we do mathematical models, like finding analogies is one thing. So there's this analogy between like oscillations in brain and oscillations in slime mold. But then one thing is like the information sharing would be cool to be able to model somehow. How do the slime molds actually, when they fuse, share information? But I don't know exactly how to do that. But I'm interested in thinking of like, we have one network with one frequency, and then we have another network with some other frequency or whatever, and then like what happens if we combine it? How is the information transformed? And another thing is like decision making, understanding how they make decisions, not just how they like form optimal solutions, but also like they make decisions between food sources. So these are like some of the cognitive abilities that would be interesting to model, but I'm not sure how to do it yet.
Thanks.
Okay. And yeah, I also had a question about the oscillations. So it seems from what you explained that the oscillations in your model are introduced through the source and the sink.
Yeah.
But in the experiments with the slime molds, and when you can observe them oscillating, is it understood what's driving those oscillations?
No, and I guess that's like one of the reasons to do mathematical models, but also one of the reasons to be cautious about exactly what conclusions you make, of course. But that's a very good question. I think like, so, I mean, when you look at this video, it's of course oscillating on the, you see oscillations on the larger scale. So like the tube, how the thickness of the tube oscillates. But then there's also like oscillations happening on multiple time scales in slime molds. So for example, like the signaling proteins in slime molds oscillate. The cell division cycle oscillates in slime molds. Also the food, like the actual particles in it, oscillates in this sense of like, if you look at how the food is transported in it, it kind of like goes forward and backward, forward and backward. So of course it's hard to say exactly what drives what, but like in our model, the idea is that what drives these contractions or these oscillating tubes is actually the particle flow, how they oscillate. But there's the thing is, the biology, there's a lot to discover from a more experimental and biological perspective as well.
No, because I would say, the oscillation, they come from the environment. They come from the interaction of your nodes with the environment. And not just from the sink and the source. The sink and the source are just the boundary conditions of source.
Yeah.
Are there any questions in the chat? I don't think so. Okay,
Yeah, one more question. I'm the one for crazy questions. This is very, very, very nice, yeah? But it is all classical. Do you know if there are any studies like this on a quantum network?
No, but that would be really interesting. I don't know too much about quantum networks, to be honest. But if you find something, please point it out to me. It would be interesting to see. But do quantum networks oscillate then?
Yeah, they do. But I have a much better idea. Why don't you come and visit us, and then we do that?
Yes, that would be. I mean, right now I'm going to. I'm soon moving, but yeah, I would be happy to come and visit you and do and study it as a like information in quantum network, oscillating quantum networks. That would be super cool. Yeah.
Yeah. Then I leave it to Amy to organize.
okay.
Yeah.
Okay. Well, thank you so much, Linnéa. I don't see any questions in the chat, so unless there's any last questions here, I think we can. Yeah, can we thank Linnéa again? It was very interesting.